Hello boys, welcome back to another session of online biology tutorials, and this session we are going to deal with chapter 1 of class 12th, that is reproduction in organisms. As all of us know that this chapter is included for your batch, that is 21-22 batch, and this contains around 3 marks, 2 to 3 marks in the board exam. And it is not just for the sake of exams, it is very significant in day-to-day life also. We go with the very term reproduction. What do you mean by reproduction? Production is generation, to produce, and something that is produced again and again is reproduction. And it is very significant because what is to be produced is the same species. As all of you know, the very definition of species, that species are group of animals or plants that exhibit similar characters and interbreed among themselves to produce similar animals or plants. Hence, reproduction is very significant because it is helping plants and animals to produce their own type. And once they start producing their own type, it ensures that the species identity is continued. That means that particular species is not going to become extinct. Reproduction ensures that species will remain on this earth for longer duration. Hence, reproduction is very important for every species. And it also says that through reproduction, the genome will be transferred from one generation to another generation. So, they exhibit similar characters. So, reproduction is very essential for the continuation of line of succession and maintenance of species in biosphere. So, the maintenance word is very important, and it is line of succession. The reproduction methods are broadly categorized into two types. So, reproduction can be through two methods. One is asexual or asexual reproduction, and second one is sexual reproduction. So the very name indicates that asexual, without the involvement of sex cells or gametes. We use the term gametes or reproductive cells. So if organisms are able to produce a similar kind without using sexual cells or gametes or reproductive cells, then it is called asexual reproduction. On the other hand, if organisms use sex cells or gametes or reproductive cells, then that method of reproduction by using gametes is sexual reproduction. So let's go by the first one. Let's discuss in brief about the first one. Asexual reproduction refers to the production of offsprings means children, offsprings, by a single parent without the formation and fusion of gametes. What are gametes? Just now I said gametes are the reproductive cells or sex cells. So there is no question of formation of gametes, and nor there is fusion of gametes. And to have these gametes, there are two types of gametes, male gamete and female gamete. So the male parent will be producing male gamete, female parent will be producing female gamete. But in this case, it's only a single parent. So there is no involvement of two parents here, it's only single parent. And that single parent will be able to increase the offsprings without the involvement of gametes. That is why it is called asexual reproduction. Since there is no involvement of gametes, the genome of the parent will be directly passing on to the children. Hence, they are Xerox copies of the parent. The offsprings will be always Xerox copies of the parent. Whatever the genes that are present in the parental organism will be going completely in toto to the offsprings. Hence, offsprings will show 100% characteristics of parent. Offsprings receive all their genes from one parent. So, they are identical with each other and they are called clones. So, this is normally one mark question. What are clones? So, similar appearing organisms with similar genome. Asexual reproduction is also known as agamogenesis. Agamo without gametes. Genesis means birth. Birth of an organism without the involvement of gametes. That's why agamogenesis, or it's also called agamogeny. Multiplication occurs rapidly and only mitotic division occurs. So example, amoeba, euglena, hydra, potato, bryophyllum and various types of asexual reproduction are summarized below. Let's see the various types of asexual reproduction. I go with the first one, fission. Fission means breaking, division, dividing. The whole parent body will divide into two or more than two portions. Each portion, broken part, will be developed into a new organism. So in this, there are two processes that are involved in this. One is called karyokinesis. Karyo means nucleus. Kinesis means breaking. So, what happens in fission before the body divides, like amoeba, one amoeba breaks into two parts. So, when it breaks, first it is the nucleus that breaks. So, the process of breaking of nucleus is called karyokinesis. First, karyokinesis takes place, followed by body division. Karyokinesis is nuclear division. First one is nuclear division. Nucleus divides. Nuclear division. Followed by somato, body division. So always in fission, there will be two steps that will go hand in hand. One is the nuclear division, karyokinesis, followed by somatogenesis, body division. So division, how do you define fission? Fission is a division of parent body into two or more daughter individuals identical to the parent. So basically it is of two types. If the parental body divides into two parts and each part becomes an individual, an adult one, then we say from one parent two individuals are formed, we say binary fission. So division of parent into two small nearly equal sized daughter individual cells. Normally bacteria, protozoans and planarians undergo binary fission. If one parental body divides into many fragments and each fragment develops into yet another daughter cell, that means multiple offsprings will be formed. Then we say multiple fission. Multiple fission. It is a division of parent into many small daughter individuals simultaneously. Example, amoeba, plasmodium. That's why plasmodium, whenever it enters into our body, undergoes multiple fission, we develop quickly malarial fever. Plasmodium vivax. Okay, that is the reason why we suffer a lot, because of its ability to undergo multiple fission. So this is the first method of asexual reproduction. And second one that we normally observe in a large quantity is budding. So it is the process of formation of bud. So budding is a type of asexual reproduction in which organisms increase their numbers through buds. Now the question is, what are buds? Buds are vegetative outgrowths present on an individual parental body. On maturity, these buds will fall off and develop into younger ones. So formation of daughter individuals from small external growths, projections, the bud, arising on the parental body. Example, Scypha, Hydra. In some terms, there will be internal buds also will be there. They are called gemmules. These are found in freshwater sponges, particularly Spongilla. And then we have got another very common method of asexual reproduction. So first one is fission, second one is budding, third one I go for spore formation. Spores are propagules which germinate to produce new individuals. There are several kinds of spores. Now, the spore is again, we can define it as asexual part of a parental body that generates a new individual. So it is basically a vegetative part. It is not a gametic one, gamete, no. It is vegetative part. And depending upon the nature of the spore and having whether the spore has got some organs for movement, they are being given different names like zoospores, a spore that moves from place to place. That's why zoospores, zoology. Sporangiospore means sporangium will be there. A special structure will be there called sporangium, which will develop spores. So sporangiophores, chlamydiospores, oida, conidia. These are varieties of spores through which organisms will increase their numbers. Hence, spore formation also is very, very important. Next, we go for the fourth one, we go for regeneration. This is the special ability of only some organisms. I repeat, it is the special ability of only some blessed organisms, I should say. Why am I saying it is special ability? It is the ability to regrow the lost parts. When I say lost parts, it is LOST, lost. Like for example, in our case, you take the moment we lose some skin cells due to some minor cuts, minor accidents, we get back the skin after some time. So the skin has got the ability to regenerate the lost skin cells. Similarly, muscle also can regrow. Nerve also can regrow. Bone, of course, whenever we have got a fracture, someone has got a fracture, unfortunately, the doctor wraps up the leg or hand wherever the fracture has taken place. In 21 days, the bone regrows, sets in. That means in 21 days, the bone cells are growing back. That regeneration. Similarly, we cut nails, we cut hairs, and these come back within a week. So this is the ability. But unfortunately, if there is any major organ being cut in our body, let's say a hand is cut during an accident, there is a complete loss of a leg. Unfortunately, so far, we are not able to get back. Of course, stem cell research is going on, that's a different story, but still it's a baby, and in India, it is still to get inside. There are certain parts within an organism that can regrow, regenerate in human body. But some of the lower organisms are blessed so much that they regrow the entire body. That's a wonderful thing. That is the reason why human beings, we should not feel that we are great. There are small organisms which we cannot see with the naked eye. They can grow fully, regenerate themselves. And one of the best organisms that I can recollect is the starfish. You break the starfish into small pieces, throw it back into the sea. That is the only condition. It has to be in its original habitat. And each piece of the starfish will regenerate the entire starfish. So that is the ability of starfish. Compared to us, it's in a very, very low category. But look at the ability of the starfish to regenerate the whole body. Whereas in our case, if someone makes us into pieces, are we getting back? No chance. So formation of whole body like starfish or of an organism, or some fragments like I said, nails, hairs, and in hydra for example, tentacles. When you cut the hydra, tentacles will regrow. That is regeneration. So formation of the whole body of an organism from a small fragment like morphallaxis, or the replacement of last part. So it is called epimorphallaxis. So we have got regeneration, spore formation, fission, two types of fission, binary and multiple, budding. And now we go for the next one, that is strobilation. So series of ring-like transverse constrictions are developed. Organisms looks like a pile of minute saucers, cup and saucer, teacup and saucer. So you get that series of the transverse sections, and each one is called strobila. The segmented body is called, as I said, strobila. For example, Aurelia. Aurelia is famous for formation of strobila. And then fragmentation. It is almost to the comparable to fission. The parental body breaks into two or more fragments. Each fragment develops into an organism: sponges, sea anemones, and echinodermata, echinoderms. And then gemme, just now I said, specialized structures which develop in small receptacles called gemma cups or gemme cups located in thallus. Example, Marcantia. It's a plant, Marcantia algae. Okay, Marcantia. And this in Marcantia we can see this formation of thallus. And then the other side, it is of course we already talked about the sporulation during unfavorable condition. Now here the important thing is unfavorable condition. When everything is favorable, okay, every organism will undergo reproduction, produce. But when things get the worst or not suitable, not conducive, then amoeba secretes a three-layered hard covering around itself. And on return of favorable condition, this encysted, we say cyst formation. Amoeba divide by multiple fission and produce many pseudopodiospores that are liberated by bursting of cyst wall. This phenomenon is termed as sporulation. Remember your Vijayanagara's slogan, when going gets tough, the tough gets going. Here exactly it is the duplication of Vijayanagara's slogan. That is, when things are not favorable, what does amoeba do? Normally, when things are favorable, amoeba will undergo binary fission. Okay, but when things are not good, what does it do? It develops like, for example, this is the structure of amoeba. It will develop 3 to 4 layers around it, and that is called cyst. So cyst will be formed, and the cyst will survive as long as unfavorable conditions are existing, to even to the tune of 2 to 3 years, they say. And during this period of toughness, unfavorable conditions, what does amoeba do? It makes whatever reserve food that is there will be consumed, and it breaks the cytoplasm. Remember I said cytokinesis. In fission I said body will divide. First nucleus will divide. I said karyokinesis followed by cytokinesis. Here also internally the cytoplasm will divide, and so many formations, small fragments will be there, cytoplasmic fragment. And each fragment of the cytoplasm will sooner get a layer, and the parental amoeba breaks open once the favorable conditions set in, and small offsprings of amoeba in hundreds will be released at a time. That means it's almost like a sweet revenge. When conditions are not favorable, it enhances its ability. And that's what we should learn, being human beings, from this small creature called amoeba. And finally, what about the plants? Plants exclusively will undergo vegetative reproduction. There will be vegetative parts through which the reproduction takes place. What are the vegetative parts other than flower? Everything is a vegetative part. If the plant increases its numbers through stem, it's a vegetative propagation. If the plants increase their numbers through root, it's a vegetative propagation. If the plants increase their numbers through leaf, it's a vegetative propagation. Hence, formation of new plants from somatic units or vegetative units are called vegetative propagules, such as buds, tubers, rhizomes. Now here we use this technology. Like one is the natural vegetative reproduction, wherein in nature, naturally vegetative reproduction takes place and the plants will go on increasing their numbers. That is, underground stem structures like suckers, rhizomes, corms, bulbs and tubes, creeping stems like runners, stolons and offsets, roots, leaves, bulbils help in natural vegetative propagation. Whereas using this natural phenomena, we can also increase the number of plants through artificial way. This is exactly what I have given you summer vacation homework. That is cutting. Through cutting, we can cut the stem, and when we cut the stem, I said you should not cut it exactly horizontally. You should have the slant cutting, so that vegetative bud will be there. And like for example rose, you can cut the rose stem slant, and one big rose stem can be made into 3-4 cuttings, and from each cutting you get a new rose plant. Similarly layering, like jasmine, mallige, you can do layering of this. Grafting, that is you have got two varieties of plants, you can graft plant A with plant B and you get a hybrid. And of course micro propagation, that is almost tissue culture. So we'll discuss in our next session about this. Until then, keep on studying, increase your knowledge.